I usually wear shorts, and somebody asked me why I spilled ketchup on them last night when I was eating Fritz. That's why. I am unable to move it forward. Aha, there we go. So I'm going to speak today not about the project per se, I am going to speak about it, but about the concept of open science and what the IMI might be able to do if it dreams big, as it were. And so, we have to focus with the problem that we're addressing in health. Around the world, we spend about $250 billion every year on biomedical research in academia and in industry. And we've been doing that for ages. And yet, for most diseases, we really don't even know what the fundamental mechanism is. Parkinson lived and described the disease in 1808. And we still don't really know what causes disease. And how can we expect industry to try to find therapeutics if we don't know the cause? And so I think we have that, in essence, describes the Venn diagram of interests, right? Industry needs to understand the cause. We want to, let's sort it out together. And one of the other problems in the sort of societal point of view is when we invent medicines, they're not affordable for most people on the planet. So no one in their right minds would think we're doing well, right? And so I think the IMI is one of those situations that we can try and fix things. And so a lot of the blame lies on professors like me. Okay, so on this graph here, what we've done is we're trying to map science, okay? And so the human genome provides the only example in any scientific endeavor where it's bounded. There are 20,000 genes. Everyone has a name. You can type the name into PubMed and tell you how many papers there are on each gene. Okay, so if you take a subset of the genome, and we chose the protein kinases as an example, and looked at the pattern of scientific activity, because papers equals number of people working on it equals money, right? You can find that most of the scientists on the planet publish on relatively small fraction of the kinases, which makes sense because these hadn't been cloned or discovered or sequenced yet. Okay? So that's the starting point. Now, if you remember Blair and Clinton standing there with great glee, this human genome is going to transform the world in 1999 or 2001. Science will change everyone. And so we put that hypothesis to the test, and we said, if that's true, the pattern should change. So I looked at the scientific activity of the planet, ranked the kinases in the same order, and said, what's going on later? So we looked at 2012, and most of the activity on the planet remains on the genes that were popular last century. Okay? Now, if you look at genetic linkages, they're all over. Okay, so it's not that these are the important ones. Professors like to think that, right? Because we're so smart, we don't want to pretend we're dumb and stuff, and we magically chose all the important ones. But we didn't, okay? BRAF here, when things get important or hot, they get hot. But here we have a fundamental disconnect, right? Parkinson to 1808, we don't understand, $250 billion. Well, this is why we don't understand, because we're not studying most of biology. Right? And it's the system that doesn't enable us to do this. So, as Pierre will attest, the good hypothesis is to try and solve this is that Canadians are smarter than everyone else on the planet. And so you can do an experiment now where you take the same keyword search but go plus Canada. So of all these papers in 2012, I'm going to extract only the papers published by Canadians. And look how innovative we are. And if I did it by the EU, by Germany, by Harvard, for all those out there who think all sentient life lives in Boston, it's not true. It looks exactly the same. Okay? So no matter where on the planet you are, you get the same research activity. And this is really troubling from a policy point of view, right? Because although science is global, we fund parochially. Governments fund science for economic development, for betterment of their citizens. We fund the best science in the pool, the small pool within our country. Canada funds the best science in Canada. It's the same science that's going on in America, the same science. So that's why it's really important to think beyond borders, right? And so with the IMI, there is an issue, right? It's focusing on Europe a little bit, like it's mega, make Europe great again. So, and so, I argue that this down here is where the true innovation lies. Now, we don't know if it's going to be connected to disease, but if we don't do anything down here, we'll keep working on this over and over again and put yet another HDAC inhibitor into the clinic. Okay? So one of the hypotheses we started, this was early in 2000s, that open science would be the most effective way to innovate. It would reduce the redundancy because you're sharing, and you'd be able to carry out research faster and better. So let me tell you what we did over the last 15 years. First, though, describe what I mean by open. Okay? So open data and open publishing, of course, are part of open science, but that was cutting edge. Remember the Bermuda Principles and the Genome Project? 1996, they decided to share data. So we're talking about sharing data, and we're so, think it's, wow, we share data. That's 20 years old. Okay? I think we should move a little beyond that. And so what's use to the data, I argue, if you can't repeat the experiment? And what you repeat the experiment with is the materials that you did the experiment with. Currently, they're shared under legal restrictive agreements. So that's no good. So we got rid of those. That needs to be happening. Preferred access to knowledge. Clinicians, like we talked about in the last two talks ago, when clinicians collect their samples and, we can't publish until I have the right to publish first. Right? These samples are human flesh that came from somebody. It's kind of odious, right? And then hospitals that have biobanks and want to monetize them, they're peddling flesh. Some sociologists should write about that. That's just gross. I'm serious, okay? Sharing data prior to publication when possible. I think everyone should try to do that. This publication as a metric, it has to go. It's our fault again as professors, right? But it has to go. And finally, we've had great success in declaring a patent-free zone. Right? So what happens outside the zone doesn't matter to us. We can't control it. But our project has a box. If you're in the box, you're not allowed to patent anything. And therefore, you're allowed to share stuff pre-patent, so you're not worried about prior arting yourself. And knowledge can really share a lot faster. So within that context, the Ultra DD project is part of the IMI, which involves our organization SGC. It's really wonderfully managed by Niels at Novartis. He does a superb job, and Michael Sundstrom in our organization. And so the general concept of all these projects that I'm going to talk about is a concept that there's a commons where there's common interest between industry and academia to discover about the unknown proteins and genes. And to do that, you need materials like structures of the proteins, the drug targets, chemical inhibitors of them. They're one of the most powerful ways to uncover biology. Antibodies to study them, screening tools, cell-based assays. The chemistry part, though, is really interesting because the true experts on the planet are in industry. So you need to collaborate with them to be able to make industry standard molecules to give away. Then these reagents go into academia and create more knowledge about the unknown. And then anybody, industry or even small companies or academics, when the knowledge is sufficient about a target and their comfort level is satisfied, they can go ahead and start a proprietary program. We don't care. Okay? And so this was the concept behind the SGC, and I'm going to show you how it sort of evolved over 15 years. It first started with three-dimensional crystal structures, where we'd solve the crystal structures and put them into the public domain without restriction. Every 20 or 30 times from each company every year, they phone us for the reagents to reproduce these structures internally. They're very commonly used inside of industry. Then 6 years later, we decided to use the structures to make small molecule inhibitors, industry standard ones, so-called chemical probes, and give them away. And the rationale behind this is if you look at every novel target in the human genome, every new protein, always, if there's a chemical inhibitor of it, that's the number one cited paper on that target in all of biology, all of the literature. It's the most effective way to learn about a new protein is to make a great molecule. Again, industry has the skills to do it, so you need to collaborate with them. But that's the purpose of that. The Ultra DD project said, well, these molecules are only good if you put them in the most relevant system, and that's in patient cells, not in 293 cells, which were made in 1996 and have evolved to be Frankenstein things. But the patient cells, of course, now you're interacting with hospitals, it becomes more complex. Again, that's where the open science comes in. You declare the zone, companies, academics, hospitals, no one's allowed patenting anything. Everyone gets to see the data as it comes off, industry, and it creates a nice box in which to play. And that project, I'll talk about outcomes later. one postdoc in the lab, and this is a pilot project for every patient foundation we interact with, she's putting her lab book online every Friday with a short little précis at the top, which in lay language, and then her detailed scientific experiments. With the idea was, why should we not share? I mean, we're funded by the public, our salary's paid by the public, we owe it to the public to share our science, not wait till we publish and put it out. So she's done that, and I'll give you some outcomes of that. That's also part of the IMI project. We've worked with the Montreal Neurological Institute, which is a hospital within McGill University. It has declared as an institution to be open for 5 years. It won't file patents on any output. It'll share its patient and clinical data, privacy taken care of, with anybody. And again, that's the concept of an institutional, getting rid of the institutional barriers to collaborate, which are the tech transfer offices. And their hypothesis is that by getting rid of those tech transfer offices, they'll get more industry collaborations. We have lots of the companies in our consortium are sharing advanced chemical inhibitors with us so we can make so-called chemogenomic sets. I won't talk about that too much. And finally, we're going to start taking some of the molecules we invented all the way to clinical trials and to the market in the open, and I'll talk about that later. So most importantly, what are the outputs? So in terms of structural biology, we and our open friends have responsible for 13% of all the human structures done ever on planet Earth. The chemical probes that we've made, we've shipped out 10,000 samples that have been used in 3,000 publications over the last 6 years. The hospital network has found one, this is at Karolinska through the UltraDD project, has found one indication for a new drug, and a pharmaceutical company was so enamored by the model that it came in and gave an extra $2 million to set up a tissue, one of these cell assay platforms in a different disease. Rachel, who's the postdoc, she's a very brave young lady, sharing her lab notebook because she, no professor or student, well, I might get scooped, but she's got guts. But she's had 7,500 visitors to her site, and what we hoped would happen, 15 new collaborations have emerged from people looking at her site from, oh, I see you've got purified Huntington, can I have some? And they talk and collaborate. It's fun. The Neuro also has 2 pharmaceutical companies came in the last 2 years to collaborate on the basis of their open science. And again, these are no patent collaborations, right? No one's allowed to patent. Genomics libraries, 9 companies have donated over 1,000 compounds. These aren't screening compounds. These are compounds, they've done medicinal chemistry, they've characterized them, there's no use for them internally, they share them with us. And the company we founded, I'll talk about that company at the end of the talk. From academic outputs, these industry partnerships don't detract. We publish 2.5 papers every week. Quarter of them are in high-impact journals that all professors like. We form new companies because, interestingly, when you work with companies closely and openly, you learn what the market is. You learn what their gaps are much more efficiently than you're a professor imagining what the company wants. And so that interaction is a really interesting one. And you don't need patented intellectual property to start a company to address the gap. You have IP, know-how, right? And what another unexpected advantage of the openness is that the science is more reproducible because you have everybody looking at it. And so Nature asked me to write this thing, I think it was last year, 2 years ago, on when you work with industry in the open, the science is reproducible. So what am I doing on time here? Okay, so I'm going to tell one story about the chemistry, and this is a story that is a pharmaceutical home run. So we had the idea in 2005, Tim and I, about why don't we start sharing these compounds? These compounds, when you make them, everybody in their plan will think, oh, watch out, you should protect it because it could be a drug someday, but it almost never is. It's way more powerful by in the public domain discovering biology. So he said, why don't we start a focus project to make tool compounds, chemical probes, industry standard, and all these, the target product profiles really kind of, for the industry guys out there, it's really kind of what's high bar, shall I say? And let's put them in the public domain. And the first example was a home run. It was this one where Panagis, a postdoc in our lab at Oxford, and Steph in Steph's lab collaborated with Jay Bradner, who's now at Novartis. He was at Harvard at the time. And the story is really interesting. So that's the molecule we made and we gave away. it's not our discovery. It's Glaxo's discovery in Mitsubishi. They deserve all the credit. They internally had found an inhibitor of one of these proteins that was undruggable, and they were trying to think, is it an inflammation? Is it for swelling? And they said, no, it isn't. So they didn't really know what to do. So GSK was daring and said, well, why don't we work with the academics and see if we can find really what's going on? So the humongous open science step was made by the inventors of this idea, which was the pharmaceutical scientists. They're beautiful experiments, and they had not been published. So they shared it with us, and we said, oh, we need to find a clinician who has access to the patient samples. So Jay Bradner, it was a doctor, and he was looking after a patient sample, a patient group that had the exact disease we wanted. So we started a collaboration. It took a little time for me to convince Harvard to be open, but it worked. And we worked hard and published these papers. This one, and Glaxo published their companion paper for inflammation. Remember, Glaxo was doing nothing with it. As agreed, we gave the compound away to hundreds of labs, and they, not us, used the compound to say, hey, you know that protein? It's important in cancer, big cancer, multiple myeloma, amylogenous leukemia. Glaxo then goes, dope, right? And says, we got a cancer drug here, and started a clinical trial 2 years and 2 months after the first disclosure of their molecule in the patent literature. That's incredibly fast. And it's interesting to contrast it with Gleevec, which is a very similar disease, a chromosomal translocation where the compound targets one of the members. Gleevec took 6 years. And I phoned Brian Drucker, who was the clinician who championed that, and he said, indeed, many years of this were failed legal negotiations, right? And so this is a beautiful molecule, and it was a pioneering drug at the time. This is a classic in drug discovery, but it did take 6 years from first disclosure in the literature to first in man. Open, it's not all due to open. Remember, there's no internet here, but it is fair to say that open helped. So what you can ask, now you're put on your GSK hat, don't give away the knowledge. That's stupid. We have valuable intellectual property. And this is a pretty good compound, right? But they said, let's do it. And they beat everyone to the clinic, and they're furthest ahead. And right now, did disclosing the chemistry stop innovation? No. There are 40 clinical trials, whole bunch of companies working on this exact same trial. It's easy to make another molecule once you know the target's cool. Okay? So this is, and we've got 50 of these now, and they're out there in the world doing their jobs of these kinds of molecules. Right? And the key step for those non-chemists is putting a molecule like this into the public domain. It costs about $1.5 million to make. Is something that not many people would do. Okay? So the future is, of course, what I would like to suggest for us and maybe for the IMI is for certain diseases to think about making medicines in the open. Right? And to do this, we founded a company called M4K Pharma, Meds for Kids Pharma. And it's going to use open science and target rare children's diseases that just don't fit the financial model of today. If you could hold your nose and raise venture capital, you'd have to charge an immoral amount of money for the drug. And so it just doesn't work. And so it's a virtual company that's going to aggregate and align existing investments around the world in academia and align them in a professional drug discovery process and commit to make the medicine affordable. We'll share all the scientific results with the community. We've got a big legal team working on this, but we'll keep the rights to use the data for regulatory purposes for those people who understand that so that we can maintain our market exclusivity. I think it's a cool thing. It balances open science and disclosure of all the data, so no one needs to repeat what we do. Everyone gets to critique what we do, but we still have market exclusivity, albeit shorter market exclusivity than we would have if we had patented it, but it's still enough, especially if we aggregate resources. The CEO is Owen Roberts. He just joined. He's a friend of mine, but he led another company that I founded and a very successful exit. So we're going to start focusing on this brain disease called diffuse intrinsic pontine glioma, DIPG. It's uniformly fatal. The children die within maybe 12 months. There's no treatments. It's in a part of the brain near the pons, and so it's inoperable because that controls core bodily functions. And it's really sad because the children come in, they don't see something in their eye, and the clinicians have to tell them that in a year they're going to be dead. Well, the parents, because the children are little. Okay? Interestingly, one of the mutations in this, one of the subset of genetic mutations, is a dominant mutation in this ALK2 kinase, which is one protein that we happen to be an expert in at our lab in Oxford. And so we've moved forward with this. We've raised funds from foundations. We've got a potent and selective series already that's brain penetrant. We've got a backup series for those who know what that means. We're optimizing it to improve the drug properties. And the chemistry program is also available for some other genetic targets that are linked to the disease. So I'm really excited about this because I think we've got ourselves a business model that will allow us to share the science and reduce all the redundancy, take a molecule to at least proof of concept and maybe to registration, and all while not taking any venture capital money and then ensuring that there's price restrictions when we make the medicine. So in summary, I would say that open science leads to competitive science outcomes for the professors out there. It leads to more reproducible science, that's for sure. It builds trusts with public and patients. In this messed up world now, it's one of the best times to be Canadian, honestly. This kind of science sharing really does reach out to different parts of society. It reduces redundant research. For professors and academics working with industry, they're so smart, right? They have so much knowledge, we never get to see it. But in interacting with them, we learn what problems they're having, and we can design technologies to tackle their problems. Pharma has contributed, I would say, about $200 million to our organization that never files patents over the last 15 years. So I don't think you can say, oh, pharma will never do it. It's just not true. And then maybe in the future, open drug discovery will maybe not only for the rare pediatric diseases, but maybe we can apply it to more parts of the drug discovery process because everybody knows the model is not working so well and the prices are going to kill our healthcare systems. So, and that's the IMI project that we're happy to co-lead with Novartis and a bunch of other companies. Thank you.
So we're going to move to the question and answer session. Are there any pressing questions for Aled before we move to the... so please. So wait for the microphone.
I can repeat.
So I understood that this... excuse me. So I understood that this molecule was never... there was never filed a patent for it by GSK. What then is the motivation for the company to do such a lot of development?
So they had IP on other molecules that they took into the... So GlaxoSmithKline, sorry. So for that, the molecule that they're... Let me back up again. If you have a protein target, many compounds will inhibit and meet the quality criteria, right? You take one of those molecules to discover what the protein does and which disease it's linked to. Then companies can make other molecules and file patents and do traditional drug discovery, which is what all those companies did. Our molecule was, as it were, a throwaway molecule to the public good to enable scientists around the world to learn more about the biology, and that pharmaceutical companies looked and said, oh my goodness, we're in. And they started internal programs in the traditional style. So it's a win for everyone. We got the paper, right? Panagus got a fellowship, Steph got a professorship, right? We all win. But all the companies are working on a target that they never knew about before. So it's a happy story, I think. And it's fast, right? If it makes a drug, and it may never do, it'll happen 4 years faster than it would have under traditional way. And so if it's a drug, 4 years of patient help.
Are there any? There's one more here.
Hi, I am Teresa Rosario Jansen. I work for UCB. My question is, are there any learnings that you had that you could now translate to pediatric cancers, downstream problems like how do you study them? I know that wasn't the focus of your work necessarily, it's more on drug discovery, but with the recent passage of new legislature in the US, for instance, with the requirement to study pediatric cancers, how can you help?
So again, You're right, our focus is drug discovery. But this project grew out of a Stand Up to Cancer grant we have with 4 clinical institutions. It was the only Stand Up to Cancer grant that had this open idea. And what we're imagining is that, and what we're doing is that the clinicians with whom we worked before that care for these children and are the geneticists, that they're the ones that I think will enable us to get the patient histories, which is a year, right, for these ones, but for other longer term follow-ups. I think it's a really neat way because I'm getting off topic, but the only owner of M4K is a trust we set up. None of us have any economic interest, so it's a real clear line. We're doing this for the common good, and thus it really invites more people to participate. You know, professors are like kindergarten children. We're all friends unless somebody gets more than us. But if nobody gets anything, we're happy to be, you know, peace and love and share. So that's the kind of structure that we set up.
And I think one final comment from Sally.
Thanks for your presentation. So I think you are in this business for some time now, right? So very passionate and successful. And the SCG is existing for some time. So my question is, what Ultra DD has delivered to you? What is the added value? Because is it a continuity to what was already done?
What delivered to me? Sorry.
Yes. What is it to you or to us, to all of us? What is the IMI project?
So the IMI,
what is the added value?
Yeah. So the IMI project enabled us to get Karolinska Institute and Oxford clinical centers to agree to allow us, industry and us, to put a team in their clinics to be able to access patient cells from the clinicians and develop cell-based assays based on the patient cells through which we could profile our compounds. Okay? And so the Ultra DD project is specifically about creating this network of hospitals where the rules of the game are no patents. We get to run the assays, not their graduate. You don't want graduate students running assays because if it's bar night, I think the cells can wait another 12. So you need to do it properly. So they allow us to go into the lab, put a professional team in place and run it. And it's interesting, it's started to bring in even more hospitals and even more companies into that sort of Ultra DD framework. That, I think, is the most exciting part of it.